all the assignment link in the Putra Blast. So any of you, any of the first assignment has been completed, then you can start to submit. Yeah. Okay. And remember, the second assignment is due 5 days after your final exam. So the final exam, I think everybody knows that is going to be open book, but in order for you to answer, you must understand all these terms. So since from the beginning, okay, from the beginning of our discussion, okay, so I hope that actually today it will be an interactive section. But then, okay, I was hoping that you can answer the question, but I think you need time to prepare. But this is basically what we have discussed. The figures that I'm showing right now is the one that the basis of biosensor or chemical sensor. And of course, this biosensor is not actually trying to replace totally benchtop machines like HPLC, like GCMS, yeah, but they are a good alternative. So, because we know that benchtop machines are very expensive. So not all at all conditions that we can have access to the machines. For example, if we have like remote area, and then we have pandemic, we need something that is portable, that is, even though it's portable and simpler, it should be reliable to 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 have a detection for something. It's not necessarily medical related. It can be, for example, you need something to test water. You need something to test if there is a gas leaking in certain industry. We heard about ammonia. We heard about toxic gas. So those kind of sensors are very important because it give like early warning, so people can be vacated as soon as possible, including the tsunami. Tsunami also has some sensors. It might be based on physical sensors. Uh, those things are real, something that you need. Yeah, but biosensors and chemical sensors, especially biosensors, most of the time is related to medical, but still we can use it in agriculture, okay, to detect the spread of disease in plants. For example, we have a lot of issues with paddy, rice field, uh, paddy plantation. There's a lot of pesticides. There's a lot of diseases that attack the rice plant. Uh, so these are the things, I would say probably not a common practice yet in Malaysia. But in future, if we are to do like smart farming, how much you want to control fertilizers, how much you want to control pesticides, requires sensors, because we uh, uh, yeah, it's difficult to rely on human resource, okay, all the time. We cannot rely on human all the time to do the checking, to do the analysis on site, or bringing in the samples to the lab. So sometimes we have to put sensors on site and monitor in headquarters or remotely from the lab. Okay, so these are the application of sensors, regardless if it is physical or chemical or biological. Yeah. So from here, you have to ask yourself, okay, if you are revising the subject, you have to understand first and foremost the definition of analyte. Okay, so of course I'm not going to recap back, but you need to ask yourself all these questions, yeah? Because if you don't understand this term, you will not be able to answer the final exam, even though it is open book, okay? So what is analyte? Okay, you should answer. And then, of course, once it is you have known, you have identified the analytes. We have also have discussed receptors. Yeah, sometimes receptors is known as chemical receptors or bioreceptors. Sometimes also it is known by other terms. Yeah, but I prefer use receptors because that fits the. If you look at the picture here. The interaction should be very specific between the analytes and the receptors. But of course, there are a lot of issues uh, in terms of specificity or selectivity, okay, and sensitive, which relates to sensitivity. So when we talk about receptors, it is very important to check on the selectivity first, because if you don't have a very selective receptors, then the sensor itself will suffer from interferences from other foreign substance that is also exists in the samples. Yeah, because we have to know that the samples contain a lot of other things, and if these receptors are not selective towards the analytes, meaning that it can react with other foreign substance in the matrix sample, then it will defeats the purpose. The whole sensor will not be workable. It will suffer a lot of errors. So receptors, we have learned about DNA, uh, antibody, aptamer, okay? And Dr. Helmi also talk about enzymes. Okay, enzyme is very useful, even though it is what you call uh, quite an old technology, but then enzyme is actually still very useful. The fourth one we learn about CRISPR. So CRISPR is actually related to DNA as well, just like aptamer. So it's just that it has moved to different technology. Uh, Not to say different technology, but I think it's like an innovative uh, enhancements of DNA related technology. Okay, so there are other receptors as well. Okay, so for example, okay, I just mentioned here that you have to take notes. There are certain metal oxides, yeah? There are certain chemical reagents that can also be used as receptors. For example, ionophore. Okay, so I've not discussed ionophore. So ionophore is a chemical reagent. Okay, I put it here. Okay. So ionophore is a chemical reagent that is specifically synthesized, yeah, to be selective towards analyte, for example, metal ions, has known some being synthesized for ammonia, ammonium. Okay, so it is chemically synthesized chemical reagent to have a selective uh, selectivity towards the analytes. Other than that, there is what we call as molecular imprinted polymer. Okay, so this one, probably some of you who uh, registered in 5407 separation that I don't remember the full. Okay, so this is example of chemical receptors. Yeah, the polymer is designed specifically, trying to specifically to mimic the analytes. So we don't discuss this, but you need to know. That's why it is open book exam. Okay, and there are, apart from enzyme, you will find metal oxides. Some metal oxides can behave like enzyme. Okay, so there is technology, there is research done, because as we know, enzymes are not robust. Yeah? It has to like be kept in certain temperatures, um, certain pH. So sometimes, and some enzymes can be very expensive. So there are certain metal oxides you can try to find out, okay, that can behave like enzyme. Okay, so these are also considered chemical receptors. Yeah, so they specifically, for example, some metal oxide can specifically react just like glucose oxidase. Okay, it can oxidize glucose into gluconic acid and further uh, being translated into readable signal. Okay, so these are among the things that you can read through that give you idea apart from DNA, antibody, enzyme, aptamer. Okay, and this imprinted polymer, I think for those of you, some of you in class in separation, you know this molecular imprinted polymer can be designed uh, serving as plastic antibody. Yeah. So antibody is biological, but uh, polymer, these types of polymer can be designed to be as plastic. Plastic means is synthetic antibody, which is good because we know also, as I mentioned in the discussion before, that antibody is difficult actually to produce. It can be very expensive in preparation, as well as it has like batch to batch variation. Yeah, it can the performance of the antibody can be different for different batches. Why? Because it is harvested from from animals such as rabbits, mice. So synthetic antibody probably can be a better alternative. So watch out for these technologies of receptors. Okay, there's a lot others that probably I also don't know. Okay, and the third part is transducer. So these also have a lot. So we have discussed about optical, electrochemical, which I can uh, discuss. There are other transducers that I actually not capable to discuss. For example, the metal oxide in enos. This is not really my expertise, but I have tried to discuss, and I hope that you have listened to the recorded video in the Putra Blast. There are also what we call as field effect transistor, FET. Okay, so those are also transducers. It looks like electrochemical, but it is not. It is based on voltage and resistance. And there are other as well called piezoelectric, SPR, surface plasma resonance. Okay, so those are transducers that transduce the interaction between the analytes and the receptors uh, to a measurable signal. Okay, so transducers are very important in biosensor development. Okay, number 4 is probably something um, that I didn't discuss, but it is very important. Okay, so you have to read this. What is biomarker? It can be chemical marker or it can be biomarker. So what are these? Okay, these are indicator. Yeah, as you can see the word marker there. It is an indicator. For example, if you dengue infected. Okay, what is an indicator inside the blood that give you a positive um, of dengue infected? Okay, if you go to the hospital, sometimes you just check your platelet numbers, right? They drop, but they can be an indicators not just to dengue. It can be malaria. It can be other viruses as well. Because once you have infection with virus, there is an effect on the platelet reading. So it's not really conclusive that you are dengue, for example. You are dengue infected, but there are certain proteins that exist in the blood once you are infected with dengue. So, for example, NS1 protein. So there are certain proteins, for example. So that is a biomarker that indicates you are dengue infected. Okay. So same goes to, for example, COVID-19. There are certain biomarkers that exist in the saliva, that exist in your throats, that indicates that you are positive for COVID-19, as well as cancer. There are certain cancers, yeah, not all. Yeah, there are certain cancers that researchers has identified a conclusive, yeah, markers in the blood sample, probably, yeah, that really indicates is a cancer. Okay, so there are of course common general marker, but some cancers has very specific biomarker. So these are terms that are very important in development of biosensors in order for you to develop one. Okay, so 1, 2, 3, 4. Okay, but of course the fifth one. So these are what we call as supporting issues that you have to identify, because I think you have done the first assignment. Most of you have done that. Uh, you have to think about the sensitivity, the selectivity. Okay, so how much sensitivity, how low you can detect. Okay, how selective is it? Whether you can detect all types of, for example, uh, I forgot. You have COVID, and then we have this respiratory that caused by a virus as well. Even the COVID also has H1N1. Sorry. There are before this we have H1N1, H1N5. I think a series of different viruses. So sometimes it is important to be very specific. Sometimes it's not. Even for dengue, for example, there are four common serotypes. Okay, and it is not important for you to know. Actually, you get serotype 1, 2, 3, or 4. But there are certain conditions, certain disease that it is very important for you to know specifically. Why? Because probably the treatment is very specific. Okay, so selectivity wise is very important in in sensors. So you have to think about this apart from the first four. And number 6, you have to also very well versed because about the surface chemistry. Okay, so this one I hope that you studied the surface chemistry, especially we have discussed a lot on amide chemistry, but there is also what we call as click chemistry. So click chemistry, there are other chemistry as well, but you need to study these two. And you have to make sure that the surface, because it is surface chemistry, you have to make sure that the material, for example, if you're using gold, if you're using silicon, what are the steps of chemistry involved, needed to be used to make sure that you can attach the DNA, you can attach the antibody and whatnot. Yeah. So this is part of the, when you work with transducers, when you need to attach the receptors on top of the transducers, we need to use a sound chemistry. Yeah, you cannot just like put them on top and expect them to work. Yeah, so this is, and the chemistry also must be suitable for the kind of material that you choose. For example, as I mentioned just now, whether you choose gold, whether you choose silicon, or other surface or carbon. So it has to suit the surface. Right. Number 7 is of course a very important, because now um, people tend to do what we call as multiplex. Yeah. Okay, let me just put the word there. So what is multiplex is, for sensor, we want to know many diseases, for example. Many analytes can be measured in one go. Yeah, it's not really practical if you have to use different sensors every time. So you meaning that let's say you have a glucose reader, so you check your glucose one, and then you want to know your cholesterol, you have to use another one. You have to withdraw more bloods, and then you have to, okay, let's say I want to know triglyceride, so I have to like punch again to take blood to check the triglyceride. So you want to know all in one. You want to know the glucose, you want to know the LDL, HDL, triglyceride at one go. Okay, so this is called multiplex. So in order to do this, it's quite difficult if all the things are just manually done. It has to be assisted by a software. Okay, for example, recent now is AI. So this also requires additional uh, expertise, yeah, where the AI can be trained to evaluate the readings, so the signal generated can be uh, produced, telling you many informations at one go. So you don't have to like do a lot of analysis just to get the reading one by one. Of course, this can be done by benchtop machines, like good machines if we have HPLCs. But imagine that you don't have the resources. Okay, so this is a good alternative. Okay, I think I'm done. So any questions? On the assignment? You okay?